the ECG signal results in PQRST wave. So why does it look like a PQRST wave? Why does it not look like sinusoidal wave, for example? Yeah, so there it will be learned in the first and second chapter. We will learn that in first and second chapter. Okay, so in week 1, we will be studying this overview of the course and the role of sensor in biomedical instrumentation system. Terms and rule for sensor and measurement course, overview of medical instruments such as ECG, EEG, glucose monitoring system, and the role of sensor and transducer. Then we start maybe either the second hour or tomorrow, definition and model of biosignal, historical aspect of biosignal, classification and biosignal monitoring. So this will continue then next week with hopefully related to biosignal and related physiological phenomena. What occurs in the cell? So hopefully you already learned about cytology and histology, right? So we will review again, and how does the things that occur in the cell originate or create biomedical signal form. So many signals like electromyography, electroencephalography, EEG, neurosignal comes from the cell level, yeah, from the neurons and the receptors. And what happens in the muscle, what happens in the heart, in the circulatory system, respiratory system, then vital phenomena and their parameters, and the parameter behavior. And this will be in week 2. And at week 3, there will be introduction and about sensor itself, either week 3 or week 4. We'll see later, because the discussion on biosignal sometimes could be a little bit wide, yeah. Quite long sometimes. So after we learn about the signal in the body, where do they originate from, how do they originate, how are they formed, then we learn how to capture them. And we learn the basic of sensor. What is sensor? What is the characteristic and terminology of sensor? And measurement. We learn about accuracy, precision in measurement, resolution, sensitivity, non-linearity in sensing and measurement, distortion during measurement, repeatability, noise, output impedance, grounding, instability, drift, and hysteresis. So why is it? Why do we need to study all this terminology? Because when we do measurement, when we measure, yeah, the biosignal, how can we know that the measurement is done properly or not? How can we know that we have measured well or not well, not good? Okay, so we need some figures of merit. We call it as figures of merit to assess and evaluate our measurement and evaluate our system, especially the sensor. And these figures of merits are in this basic terminology, yeah, accuracy, precision, resolution, etc. And further, this will be followed by dynamic figures of merit, such as time response, bandwidth, then some other figures of merit and biocompatibility, because we are speaking about biomedical sensor. So some sensor, for example, needs to be attached to our body. Yeah. If it is attached to our body, it means that it has to be biocompatible with our skin. Some sensor need to be implanted inside our body. It means then it has to be biocompatible with our tissue. There should not be, for example, rejection from the surrounding tissue towards that implanted sensor. Then some discussion on the microfabrication of biomedical sensor. So with the advent of microelectronics, yeah, besides IC, integrated circuit electronics are made smaller and smaller, and then so that we have our smartphone, our laptop, etc. Sensors are also made smaller also. Yeah, and we will hopefully have some time to discuss about how some sensors are made small so that they can be easily integrated into, for example, wearable system or into portable system, and even some of them are implantable inside our body. Then after that, we learn the individual type, yes, of sensor based on the energy domain that they are working with, or information domain. What information are they tackling? For example, in physical sensors and measurement, the introduction of resistance sensor, it means sensors that use resistive principle, yeah, like strain gauge, thermistor. Maybe you learn some of this in the first 3 semesters in your study here in biomedical engineering. Already in electrical engineering or in electronics, you use some of the sensor for your projects. Yeah? Thermistor, for example, like sensor that converts temperature difference or temperature increase or decrease into resistance change. Then inductive sensor and measurement, sensors that use the induction principle to detect various kinds of physical measurement. Measurement means parameter being measured. For example, inductive sensor can be used to measure rotational speed or translational speed also. Then capacitive sensor and measurement. Sensor that uses capacitance or capacitive phenomena to measure other uh, energy or information form. Then piezoelectric sensor and measurement. Sensor that uses piezoelectricity to measure things, to measure physical or even chemical phenomena. Magnetoelectric sensor and measurement using magnetoelectricity phenomena that convert magnetic field into electric. Photoelectric sensor and measurement. The sensor use photoelectricity to convert information in the form of radiation, yeah, photon radiation, into electric parameters like voltage or current. Thermoelectric sensor and measurement. These are sensors that use thermoelectricity like Seebeck law, Seebeck phenomena, converting temperature difference into voltage. Then we study pressure measurement, various pressure sensor. How are they being applied in biomedical measurement? Yeah, for direct pressure measurement or indirect pressure measurement. Then flow measurement, how to measure biomedical flow measurement. To measure, for example, blood flow, tissue blood flow, respiratory gas flow measurement. And motion and force, this is especially important in biomechanics subfield, yeah, within biomedical engineering. Yeah, measuring motion to measure motion, to measure force. Then temperature, heat, flow, and evaporation measurement to measure temperature or heat flow. After that, we will learn bioelectricity and biomagnetic measurement. Yeah, so this is one of actually the most common biomedical signal we want to measure. Yeah, ECG, EAG, EMG, EOG are examples of bioelectricity measurement, where usually we use electrodes attached to the skin or in the corresponding organ, yeah? Or sometimes being not touched to the skin, but being penetrated into the tissue using needle type electrode to measure the potential of that body, and then to measure the potential difference between different parts of the body. So this is bioelectricity measurement. And there is also biomagnetic measurement, even though it is not yet common yet, but maybe will be more common in the future because in the biomagnetic measurement, we do not really need to have invasive measurement. Yeah. MRI is actually one example of biomagnetic measurement where the magnetic field is being applied in surrounding our body, and then the response from individual spin of hydrogen spin in the water molecule within our body is being monitored. So after that, we will cover chemical sensor. Yeah. As we know that most of our body consists of chemical compounds with water, and then that water in blood contains a lot of metabolites, which are basically chemical compounds, protein, what else, hormones, and small molecules, gas molecules, etc. So how to measure them, how to analyze them? And this is can be measured using chemical sensors. So we will study ion sensor, gas sensor, as we often see recently in the news. Genus, researchers from Universitas Gadjah Mada developed, for example, genus that can detect or diagnose COVID-19 from the breath of the subjects. How can it be done? How is it being developed? So we will study from the element first, the gas sensor, and then study intelligent chemical sensor array. Genus is basically an example of intelligent chemical sensor arrays. Then optically based chemical sensor. Then we will study about micro total analysis system, where we include microfluidics and the processing of the sample inside the sensor itself. Then about sensor networks, where different sensors from different places are connected through electronic or computer networks, and then the information can be processed simultaneously. Measurement by indwelling sensor, X5O measurement outside the body. Transcutaneous measurement at the subsurface of the skin. After that, we will study biosensors. Biosensors in this case are defined as sensors that use one form or different forms of biomolecules, to conduct measurement, to measure any biochemical parameter. We can use enzyme, antibody or antigen sensor, tissue sensor, organ-based sensor, cell-based sensor, or even whole organism being used as the biosensor. And they are combined with physical transducer that we have learned in the previous chapters, like using cantilever, quartz crystal microbalance, surface plasmon resonance, etc. And then what are their application in biomedical environment, etc. Then week 13 and week 14 will be used for finalization of your project and assignment presentation. So as always, that my subject usually entails a project assignment. You for each for you to finish, by the end of the semester you present it. So in this sensor and measurement system, it will maybe, I'm still thinking whether the assignment will be also integrated with the biomaterial assignment or not. So you will be divided into two groups. Okay. So I have decided, for example, one group led by Ezra, the other one by Tanita Grace. Okay. And you will be continuing two projects made by the previous batch, during their biomedical instrumentation study. The first one is developing a small micro PCR, to amplify genetic, PCR using fabric-based device, using heater and thermistor. And the other project is about developing infrared thermometer using CNT coated thread, carbon nanotube coated thread combined with uh, hexaboron nitride coated fabrics. So this way the execution of this project also depends on your knowledge later on biomaterials. Okay, so in biomaterials, we will also cover many of the materials, yeah, the knowledge in the science and technology of materials. Because when we develop, what I want is for you to learn is when you develop the sensor and measurement system, you really develop from the basic, yeah, from the principle, and then processing some materials, and then trying to develop a measurement system out of that those materials and sensor system. So having said that, I understand that during this semester you cannot work fully in the lab, in the laboratory, yeah, maybe until the rest of this semester, yeah, because of the pandemic. We are not yet fully safe, even though the vaccination program already starts. So most of the projects in the lab will be conducted by my research assistant, Samuel Andrew, as well as by the lab assistant, Kak Devita. Okay, so you will be consulting with them and what to do, and they can also explain to you. And then, but the idea has to come from you. Yeah? You will be the one that is doing the literature study to back up the information needed to do the project, and then you will ask them to do for you. Yeah, of course, they can also give suggestion for you because one way or another they have more experience in biomedical in experiments, yeah, related to this. So this will be a continuation from previous semester project by, as I mentioned, your senior, which is who are mostly now in Germany. They develop this PCR as well as the infrared uh, thermometer. However, they haven't finished yet. They did until a certain degree, but not yet finished. So you will study from their reports how far they have done, what are still the disadvantages or shortcomings that they still have in their system. Then you can improve based on that. Yeah. And we will have occasionally every 2 weeks, for example, uh, monitoring of your projects, 2 or 3 weeks monitoring to your project, and that monitoring will be included in your marks, yeah, in your marks, so that the project will be included in the final marks. We'll see if the project is, if it is well executed, then we may not need to have written final exam. Okay, so I will then consider that basically based on your result in your final project, you already have mastered all the subject, yeah, about sensor and measurement. Yeah, because later during the monitoring, also during the presentation, I will be asking, for example, the figure of merits here, yeah, like accuracy, precision, nonlinearity, yeah, about the system that you are developing. If you know how to answer well, of course, why should we do another exam? But if I see that your understanding or your execution of the project is not well enough, then yeah, we may need still a written final exam on the sensor and measurement system. My plan is also, like one of the other lecturer, is maybe to have small quizzes every week, yeah, so that then you will be forced to learn the concept before and after. Yeah. So maybe either starting from tomorrow or starting from next week, because I haven't uploaded the material into the Google Classroom, so it would not be fair if I do it today, yeah, or tomorrow even. So maybe we will start next week to have small quizzes after the end of every chapter. Yeah, small quizzes, maybe just half an hour, consisting of 10 questions, multiple choices, for example. Yeah, so that then you can understand, you are hopefully eager, you will be forced to read the material beforehand, before the lecture, as well as after the lecture. Okay, are there any questions related to this?
Yes, sir. I
Yeah,
want to ask about the final project. So the final result is the product, or it's just a paper?
both.
Okay.
Why both?
Right.
Because you are making something. Yeah? It's just different than
Uh,
your assignment in Introduction to Biomedical Engineering. In Introduction to Biomedical Engineering, you are only asked to write a review paper. So in this case, you only read, and then you write your you of course you need to process your reading and then put it into review paper. So in this case, you also need to read, but you need to develop something. However, since you cannot work yourself yet in the laboratory due to the pandemic situation, Kak Devita and Sam Andrew will be the extension of your hands, yeah, in the lab. So this is similar like in the previous semester, the biomedical instrumentation students, your senior, they use Kak Sam Andrew's hands and Kak Devita's hands to build the project for them in the laboratory. We will see lah, the condition. We don't know, right? If possibly, hopefully, May, for example, well, we pray, may the condition have been much improved. Yeah, then maybe you can already start working in the lab with limited number of students at once, yeah, at one time. Okay. Otherwise, yeah, most of you will be still working at home, and then do Zoom session also, maybe with them, Zoom or Google Meet session with them, Kak, Kak Sam Andrew or Kak Devita in the lab, that they are the one doing the experiment for you. And the data will be given to you. You will be doing the processing of the data, statistical processing of the data. Okay, and then making it into good graph. So you make a report, but during the presentation also you do a demonstration of what you made. Yeah, during, of course, with the help of Kak Sam Andrew and Kak Devita during this whole semester. I hope within, at the latest by next week, I can already upload the report from the previous student group, a biomedical instrumentation group. So you can start from where they left. Not, you don't need to start from scratch, from zero. You can start from by reading their reports and reading maybe the references that cited by them, and then your group also try to develop your own idea, how to develop further. For example, the PCR group, the time they haven't, they already managed to hit up, but they haven't really finished developing the controller algorithm, the control algorithm using Arduino, for example. The infrared, um, the CNT-coded thread has been proven, for example, can transduce thermal signal into resistance change. But the infrared absorber is not yet working, for example. Yeah, so it does work as a conduction transducer, but not yet as radiation transducer. So then you can brainstorm within your group to how to solve their problem, this problem that is left by the previous group, and then improve their sensors. Okay,
okay, sir. Thank you.
you're welcome. So that will be all for this hour.